my goal, my job as a research advisor is to train students how to do research, to train them to be good researchers. That is to say, my goal is not to get publications, not to get grants. My goal is the process. My goal is to help students learn how to do research. I want them to be good critical thinkers, and I want them to have great foundational analytical and technical skills when they finish their projects. And so what I've learned is that to accomplish that goal, you need to design projects that are accessible. That is, projects that students can easily wrap their minds around, projects that students can easily explain to their friends and family. And so those tend to be projects that are really hands-on, that can be accomplished in a short time period, and that can be done on a shoestring budget. And I kind of learned the hard way that perhaps the most important thing that you have to think about when doing student research is think about what the students want to do. I spent the first 2 years here at Maryville designing what I thought were these really nice, accessible projects dealing with the genetics and behavior of speciation, because that's what I'd studied for my PhD and my postdoc. And then I couldn't really get very many students interested. And they told me that they thought it was interesting, but it was basically hard for them to get their minds around it. So I've kind of had to expand my areas of expertise to incorporate student interests. So everything that I'm going to talk to you about today are projects that were initiated by students. Students came up with questions. They came to me and said, hey, what can we do to ask these questions? And so projects have to be accessible, but they have to be student-centric, in my experience. So one of the first projects that a student came to me with was, his name was Joe Hoffman, and Joe said, what's the relationship between uropygial oil and microbes? And so for those of you that don't know, the uropygial gland is a gland that sits on the rump of a bird, and then it secretes oil. It's a sebaceous gland. It secretes oil that those birds then use to keep their feathers in good shape. And so we have this picture of this duck in our lower right-hand corner, and what she's doing is she's squeezing that uropygial gland in her bill, and she's getting that oil on her bill, and then she's going to rub that oil all over her feathers, so throughout the feathers of her entire body. And what she's doing is she's using that oil to keep her feathers soft and supple. Bird feathers are made of keratin, just like our hair is. So they use uropygial oil like we use conditioner or hair products, or I don't really have much hair to do that with, but if you have hair, then that's what you would use it for. So the basic idea is that a feather is this keratinaceous structure, and it consists of a central shaft and then some veins. And those veins have what we call barbs and barbules. And those barbules have these little hooks at the end of them. And those hooks hook with other hooks, just like Velcro does. And as long as the bird uses this uropygial oil to make sure that those hooks are soft and flexible, then they'll maintain their bond with each other. The hooks will maintain their overlap with each other. And that feather will be able to maintain its integrity. It'll look like a feather and it'll hold up like a feather. But if the feather dries out, if those hooks become dry and brittle, then they'll break. The feather will lose its integrity and it'll get holes in it. And so it will become less effective for thermal regulation and less effective for flight. So that's what we know. That's classic ornithology. That's what we know about the function of uropygial oil. But the relationship between uropygial oil and microbes is sort of an expanding area of research. And this other picture down here of this American robin rolling around in the dirt, some of you may have seen birds taking a dust bath. Birds are more likely to take dust baths than they are to take water baths. And the reason is because most of their feathers are water repellent, so a water bath doesn't really do them much good. And when they're taking that dust bath, one of the things that they're doing is trying to kick off ectoparasites. So they're trying to kick off ticks and mites. But one of the most important consequences for that dust bath is that they're inoculating their feathers with bacteria. So we know that all environmental surfaces, including soil, are just teeming with bacteria. So when that robin rolls around in that dirt, it's picking up millions of bacterial cells, and it's infusing its feathers with those bacterial cells. And so we just wanted to know at a basic level, is there a relationship between Europygial oil and preening, and then the microbes that are going to grow on those birds? So just a basic experimental setup was we would catch birds and we would swab their feathers. We would swab the soil in the environment where we found those birds. We would culture microorganisms from both samples, and then B minus A should be the species that are inhibited. So the basic idea is that we were kind of hypothesizing that there are some bacteria that would be good for bird feathers, or at least not hurt them. And there might be other bacteria that would be bad for their feathers. And so we were hypothesizing that they would want to inhibit the growth of bacterial species that would be bad for their feathers. And we're kind of at the beginning of this research. But this is Joe's work. So Joe was the person who came to me and initiated this project to begin with, and this is the outcome of his research. And so what he found was that there are some bacteria that secrete keratinase. That is, they secrete an enzyme that degrades keratin, degrades bird feathers. And it's called Bacillus licheniformis. And so Joe cultivated specifically Bacillus licheniformis from feather samples that we took from birds. And in this top graph, this is in cardinals, in northern cardinals. There's me with the northern cardinal. He did a paired analysis where he compared the feather reflectance of birds without bacteria that he couldn't cultivate bacteria from, compared to birds with bacteria. And so this is the difference in feather reflectance between those birds that don't have bacteria and birds that do. And so the higher the number, the better condition the feathers are in. That is, the more light they reflect. And so what Joe found was that for cardinals, birds that don't have any bacteria, don't have any of this Bacillus licheniformis, their feathers are in better shape. They reflect light much better than those that had bacteria. So that kind of supports this hypothesis that there are bacteria out there that a bird does not want growing on their feathers. And if they can inhibit the growth of those bacteria, then their feathers are going to be in better shape. Now, interestingly, Joe did the same analysis with tufted titmice. So this is one of my researchers, Jenna, she's got an oven bird and she has a bunch of tufted titmice. But Joe did not see the same relationship in titmice. That is to say that there was no significant difference between the reflectance of titmice that had licheniformis and those that didn't have licheniformis. And so it's a little bit of a puzzling result. But one difference between cardinals and tufted titmice are that tufted titmice have a lot of melanins in their feathers. So melanin is a structure, it's a pigmentation deposited into the feathers as they grow. And we know that melanins can protect feathers against degradation. So one possibility might be that for cardinals, it really makes a difference, what bacteria you have growing in your feathers because you don't have any melanins. But for titmice, it might not make a difference because you've got melanins to protect yourself. So this is a really cool starting point because it means that this relationship between Europygial oil and feather microbes could be different for different species. That is to say that we might see antimicrobial effects in some species that we don't see in other species. So, like I said, we're kind of at the tip of the iceberg with this research, but we have some really, really interesting results to start with. Another project that a student came to me with was why is capsaicin hot? That is to say, when we eat a spicy meal, why is it spicy? How did capsaicin evolve to be hot? And the classic example that you see in textbooks going back 40 years is that birds can't taste anything, let alone spices, and mammals can. Mammals have an excellent sense of taste. So the basic idea in the literature, at least, is that hot peppers want to be hot because they want to deter mammals, because mammals have teeth, and those teeth will crush their seeds. But they want to attract birds because birds will swallow the pepper whole, and it'll go straight through the digestive system, and they'll poop it out, 100 yards away, and now you're dispersed. So that's sort of the simplistic classic model for why peppers are hot, to deter mammals and attract birds. And then this is a picture of a wild pepper that we have growing on campus. I've planted them all. There are no native wild peppers to Missouri, but I planted them for research purposes. And you'll see that some of these peppers have been bitten through. And so I have video of a northern cardinal, coming to this pepper bush and basically snapping off half of the pepper and eating part of it. And then he just sits there and eats away, and it looks like he's chomping away at the seeds pretty good. So it didn't really look like he was eating the pepper whole and then flying away and passing it. So we wanted to look at this basic framework for how peppers got to be hot in more detail. And what we found is that birds, in fact, can taste, and they can taste capsaicin. So when a pepper is hot, they can taste it. But their threshold is different than mammals. That is to say, we might find a meal spicy before birds would find it spicy. Another thing that we found is that browsers, deer in particular, don't care about heat at all. So the hottest pepper in the world, the Carolina Reaper, comes in at 2 million Scovilles, and I can put out potted Carolina Reaper plants on the plot, full grown, lots of ripe peppers on them, and deer will come in and browse them down to sticks. Deer do not care whatsoever. Now the deer might suffer some consequences down the road. I'm not able to follow them and see what happens to them. They might experience some discomfort, but at least in the short term, being hot offers no protection against a deer whatsoever. Instead, what we found is that deer care about the plant morphology. So there are some wild pepper plants, in particular some that are native to Central America, that have a very low leaf density, and they're very twiggy. So they grow straight up and down. They're easily lignified. That is to say that they turn from soft green tissue into hard woody tissue very easily. And deer tend to leave those alone. And then the other kind of plant that deer leave alone are like this one in this picture, and they stay low to the ground. So they stay low and shrubby, and deer tend to avoid them. So at least what we see so far is that plant morphology is more important to deterring mammals than heat, than being hot, than capsaicin is. And we've done feeding experiments with small mammals like squirrels and chipmunks. And what we found is that they will eat peppers. They'll eat these peppers. Like if I pick these peppers and put them in a tray, they will eat them as long as they're not hot. That is to say, as soon as there's any heat, they'll choose to eat food that's available that's not hot. So what we have is sort of this picture where natural selection would favor peppers to be hot so that they could avoid things like small omnivorous mammals, but not too hot, because birds can actually taste things that are hot, and they actually don't like to eat things that are super hot. So here's some of the results of the work that we've done so far. So some commonalities in these two graphs where we're comparing mammals and birds. On our x-axis on each one, starting on the right, I have a picture of ghost peppers. So what we did is we took sunflower seeds, we took black oil sunflower seeds, And we prepared capsaicin solutions that were 1 million Scovilles, which is this yellow dot, which is roughly the equivalent of what you might get in a ghost pepper, 500,000 Scovilles, which would be roughly the equivalent of a habanero pepper, 250,000 Scovilles, which is roughly the equivalent to a jalapeno pepper, and then the control, which was 0 Scovilles. So we prepared those solutions. We took these black oil sunflower seeds, and we soaked the black oil sunflower seeds in these solutions of various heat concentrations. And then we dried them, and we offered them to squirrels and birds. And so for the squirrels, what we did is we put them in feeding trays. So for one experiment, we put 2 feeding trays, 2 feet apart from each other, one that had a control treatment, so seeds that didn't get any capsaicin, and then the other where they were getting one of these various controls, so either 250, 500,000, or 1 million Scovilles. And so what we would expect for mammals, because we can taste capsaicin, is that they would always prefer the untreated seeds. And in fact, that's what we find. No matter what you give squirrels, they will always prefer to eat the untreated seeds. And so maybe I should explain that our y-axis here is percent removal. So the difference between how many seeds they're removing from the control versus how many they're removing from the treated seeds. So that is to say that they're always removing significantly less seeds from these treated seeds than they are from the control seeds. And then over here we have our bird treatment. So the setup is the same, except now we're just taking those seeds and putting them in 2 bird feeders that are 3 feet apart from each other. And so we're just looking at the visitation rate. So how often the birds are coming in and taking seeds away from any given feeder. And according to the classic hypothesis, birds can't taste, so they can't taste capsaicin. So we wouldn't expect any of these to be different. And in fact, that's what we see. So for 250,000, for 0, for 250,000, for 500,000, they don't show any difference in visitation rate. They visit the 2 feeders equally. But when we get to 1 million Scovilles, when we get to ghost pepper level, then birds significantly prefer to visit the untreated seeds. So at a certain point, birds can taste it and they don't like it. And that point is just different for birds and mammals. So a group of students came to me this past summer and have proposed a new line of questioning. And the basic idea is that, with this classic model of why capsaicin is hot, the idea is that mammals have teeth, so they'll grind up the seeds, and birds don't have teeth, so they'll just swallow them whole and pass them through. But that ignores the fact that birds have a gizzard. And a gizzard is a muscular digestive organ that comes right after the stomach, right after the proventriculus. So a bird doesn't have any teeth, but they'll swallow their food whole, stored in the crop for a little bit, goes to the proventriculus, protein gets digested, and then it gets sent to the gizzard. And the gizzard is really, really effective at grinding up any fibrous material in the food. So just because they don't have teeth doesn't mean they're not grinding up their food. In fact, the gizzard can be more effective than a mammal's teeth in grinding up food. So we're kind of proposing a novel hypothesis for why capsaicin is hot. And that novel hypothesis is that it doesn't have anything to do with mammals at all. A pepper could care less about mammals, right? The morphology is more important. That capsaicin is hot to attract the right kind of bird and repel the wrong kind of bird. That is to say that if you're a hot pepper plant, you want to attract an insectivore or a frugivore, something with a small gizzard that will pass your seeds unmolested. But you don't want to attract a bird with a large gizzard that's going to be really effective in crushing your seeds. And we were going to start this project this spring. I have 100 pepper plants that I had going, wild pepper plants with small fruits that I had going, and we were going to put those out this spring and see if we could get the birds to eat them and then catch their poop and see what grows. But unfortunately, because of coronavirus, that'll have to wait until next year, hopefully next year. But the basic idea is that we've got a plot where we have bird feeders, and then I was going to put all the pepper plants out around these bird feeders so that we could get both granivores and insectivores to come in and hopefully eat fruit. We catch them, we hold them in a bag, they poop in the bag, we let them go, and then we turn the bag inside out and we scrape that fecal matter into potting material, and we literally just see what grows. And so the basic hypothesis is that the birds with the bigger gizzards should have much less growing from their fecal matter than the birds with the smaller gizzards. So, a cardinal versus a chickadee. And I'll admit that we have some experimental details to work out, but I think there are a lot of students that are really, really interested in this project, and there's a lot of side projects that we could develop from this. So this particular aspect has proven to be one of the most attractive projects that I've proposed thus far. Growing poop. So all of the research that we do, even though I've expanded the breadth of my expertise to incorporate all of this research, I try to make it such that we can all do it together and all do it in one cohesive research project in the summer. So even though I have students working on very different projects, we kind of all tie it in together so that we're all working together. And so to make that work, we have to tie in a bunch of different things together. So basically, the first thing that we have to do is have these bird feeders where we're feeding seeds and mealworms, because we want to attract granivores like cardinals and insectivores like robins. And then I need these pepper plants because I want them to eat these wild pepper fruits. So I had 100 pepper plants growing. And then when we had stay-at-home orders come through, I had to take 2 months ago, I had to take all of my wild pepper plants, put them in the ground at school, and just pray that they would not get frozen or eaten by deer. So it has not frozen since then, so we haven't had a frost. So they should be alive, but I don't know, because I haven't been able to go to school and see, but we'll find out eventually, I hope. So then the idea is we get birds coming where they can eat peppers, they can eat mealworms, they can eat sunflower seeds. We catch them, we hold them in a bird bag, we let them poop. We take their poop sample, we take them out of the bird bag, we swab their flight feathers because we're interested in specifically what's on their flight feathers. We take those swabs into the lab, we cultivate those swabs, and then we put the birds in a holding box and we videotape them flying away in slow motion. So I didn't have a chance to talk about that today, but we're also doing projects looking at variation in flight efficiency among small songbirds. So it turns out that the hows and whys of flight are not worked out within groups of birds. So we know big picture, like why eagles can fly, in a certain way, and why falcons can fly in a certain way, and why songbirds, like forest birds, can fly in a certain way. But among forest birds, those details just aren't worked out. So that's another project that my students are working on. But that's all I have for you guys today. So thank you very much for listening.
Thank you so much, Gabe. That's great. If you don't mind stopping your screen sharing so we can turn and swap over to the Q&A section, I'll pull up some of the questions that we have. And we're going on. And so my first question is that we have a lot of students who are at home right now who are interested in doing their own scientific experiments, but are wondering if you have any tips on how you might be able to do your own science experiment if it's one of the first ones you've ever done. If you want to go outside in your backyard and study something, if you have any tips on how you could approach that or make that more accessible to students doing this by themselves.
I would say go for it. So a lot of students that come to me as freshmen in particular think that research has to be something that's groundbreaking or expensive, like you have to be curing cancer or curing coronavirus. And so what I tell them is just to relax and think about things that are interesting to them, anything at all, and just scratch the surface of what's interesting to you. And what you'll find is that we know less about it than we think we do. And there are really simple experiments that you can do to test even stuff that's supposed to be like, well known and well understood. So I would say for students just doing stuff at home and in their backyard, they can actually make meaningful contributions to the scientific community doing very, very simple experiments. And start just by, asking the question and seeing what they find on Google. And even sometimes, replicating things that people have, are already done, you'll find that a lot of the explanations that you find online are not as well worked out as people make it sound. So I would say you can definitely make interesting and valuable contributions at home.
Cool. I have a question coming in from Griffin and Mari, who are watching on at home, and they were wondering if birds can control or regulate how much oil their feathers that they produce for their feathers.
That's a really good question, and that would make an awesome research project. And I would say that we don't know. Sort of our working model is that there is variation in oil secretion among individuals, but it tends to be tied to condition of that individual bird. So birds that are, in really good shape, they've got plenty to eat, they're not stressed, they're not worried about finding mates or getting eaten by predators. Those birds tend to secrete more uropygial oil than birds that are, maybe worried about where their next meal is going to come from, things like that. And so generally speaking, that contributes to the feather quality of those birds. If they're in better condition, they make more oil and they, put it onto their feathers and their feathers are in better shape. But whether or not that's a question of cognitive regulation, that's a really good question. Can a bird who's in good condition change how much uropygial oil they secrete? Excellent question.
Well, that's great. The next question we have coming in for you from a student who wanted to know, how do you catch birds? Seemed like that might be something hard to do.
Yes, it is hard to do. We have what's called a mist net, and it looks like a volleyball net or a badminton net. And let's see, birds have amazing vision, but they tend to focus on things that are far away from them. And so if you put up a mist net, it has very, very fine polyester black fibers that maybe looks like a spider web to a bird. And as long as you put it in the shade or in the dark, or you're going early in the morning, then they'll see it. They know that it's there, but to them, they're looking beyond it, and they think that it's just something that they can fly through. So they fly straight, towards wherever they're hoping to go. In our case, that's towards a bird feeder. And then they hit the net, and then these nets are designed such that there's slack in them. So when the bird hits, it falls down into a pocket, and then they can just safely rest in that pocket, laying basically on their back, and then you can come and take them out.
Neat. That's really cool. The next question that we have for you is, how did you first get introduced or excited doing your own scientific research? What made you kind of go along the path that you've gone on currently?
So that's a good question. I was a biology major, but I was a pre-vet major. That is to say, I wanted to go on to veterinary school, and that's because I loved animals. I love dogs. I love cats. So it seemed like a natural choice. And then the summer after my freshman year, I got offered an internship with the Vermont Center for Eco Studies. And that internship was working with small songbirds on a mountain in Vermont. And to me, wanting to go to vet school, I didn't necessarily see the value in living on a mountain and working with small songbirds. But fortunately, my parents convinced me of the value of it. And I went and I spent that summer living on Stratton Mountain in Vermont. And it was probably the best summer of my life. It was amazing meeting researchers who cared so much about the natural world, who cared so much about learning about the natural world. People who were, they were good scientists, which was really cool to be exposed to. But they were good human beings. They were interested in social justice and environmental justice. It was a group of people and a research subject that just forever changed my life. And so I knew from that point forward that I wasn't going to go to vet school, that I wanted to do research in some way, shape, or form. But it was just it was more or less a fluke that I applied for this internship because I knew I needed those valuable experiences, and I got this one.
That's great. Our next question is, what is your all-time favorite bird?
So I get that question a lot. And my favorite group of birds are the North American wood warblers. So North American wood warblers are birds like, let's see, so you guys are in California, right? Yep. Roughly. Mm-hmm. So let's see, I'm not sure what warblers, I've been there once, but it was in the winter, so I don't remember.
Yeah, we have a few, but not that many.
Not that many. Well, they're a very, like you get the black-throated gray warbler. I know you guys have. Yeah, they're just a really cool group of birds because they've experienced an adaptive radiation. So not that long ago, there was one species of North American wood warbler, and it has quickly diverged into 30 to 40 species, and some of them are orange, some of them are blue, and some of them are drab, and some of them are green, and they have all sorts of different ecological niches. So I really like them because they're fun to watch. They have really cool behaviors, and they're accessible. They're easy to see. They're easy to work with. So I really enjoy wood warblers. Black-throated blue warbler is my favorite, though.
Nice. About how long ago was there just that bottleneck in the species of wood warblers?
Let's see. It's been a while since I've looked at the literature, but I think we're talking 500,000 years.
Okay, neat.
500,000 years, yes.
Cool. Our next question is maybe on the more applied end from your research, but we have someone who has a bird feeder at home that they have a problem with squirrels getting into. And they were wondering if after listening to your talk, if putting hot peppers or hot pepper oil on the bird seed in their bird feeder could be an effective way to deter some of the squirrels that get into the food, but maybe not the birds that they put the feeder out. And if you had any thoughts or tips on that.
So that's a good question, and you'll see a lot of commercial products that say chili pepper infused seeds or whatever, and it keeps the squirrels away, but not the birds. What I've found is that the birds, even though their threshold for making significant choices between untreated and treated seeds is pretty high, you're not going to put ghost peppers out on your bird feeder. But even at low concentrations, I found that those products tend to keep birds away also. Mm-hmm. I guess I don't necessarily recommend using hot pepper oil or hot pepper in your bird food to keep squirrels away. Unfortunately, the most effective way to make sure that it's birds that are eating your food and not squirrels is a good old-fashioned baffle. And positioning, making sure that your feeder is somewhere where squirrels can't jump onto it, right, from a branch or from a structure. So, good old-fashioned manual, or not manual, but physical barriers are the best way to keep squirrels off.
Yeah, and for the audience at home who doesn't know what a baffle is, it'd be an upside-down metal cone that a squirrel couldn't jump around.
Absolutely, yes.
Okay. Perfect. That's great. And then the last question that I have for you comes from our teaching staff, who has to help students with kind of taking their own research project and kind of implementing it and looking at the data. We have a lot of students who go out, collect their data, and for their first round, they find somewhat inconclusive data. It's not really corroborating or refuting their hypothesis, and they want to know, or sometimes they get discouraged at this point. And if you have any tips for what to do when you go out, do an experiment, and you get inconclusive data, if you have any tips for students working through that problem.
Well, first of all, I'd say congratulations, because that's the most common outcome for any of us. So if you've gotten to that point, then you've done a good job. But I try to tell my students that inconclusive results are very valuable, because they help us decide what the next step is. And so sometimes inconclusive results can mean that, or not significant results can just mean that your initial idea wasn't right in the way that you asked it at the time. But other times it can help us inform our experimental design. Sometimes we have to do the experiment to understand that our experimental design wasn't actually appropriate to test what we wanted to test. So inconclusive or non-significant results are always informative. They always help you decide what the next step is. So I would, well, it's natural to be discouraged. They're an important part of the scientific process.
Cool. Well, that's great advice. Thank you, Gabe. And that's all the questions we have for you today. We really appreciate the time you took out of your day to tell us about all your research. And I think there are a lot of people at home who like spicy foods who are going to think about them differently now than they did before.
Yeah.
So that's great. Thank you so much.
You're welcome. Happy to help.
Cool. Take care and have a great rest of your day.
Thanks. You too.
Cool. We're offline now, Gabe, but thank you.